give either cannabidiol or this compound, which we call it HU320, it reduces the inflammation. So if we look at the clinical score of that rheumatoid arthritis I was speaking about, we see the same thing. This compound, like CBD, reduces the production of arthritis or the development of arthritis, and it is an anti-arthritic drug considerably better than cannabidiol itself. So if we look at the damage produced by these in these models of damage of joint erosions, and the numbers here amount to 100 in every case. Well, if we don't give a drug, 57% show moderate to severe degree of joint erosion. But if we give HU320 or cannabidiol, the moderate to severe erosions go down very, very significantly, and quite a bit of the animals stay normal. So, to summarize this part of my lecture, we believe that the anti-arthritic potency of cannabidiol, which is present, probably is the major component of most cannabis plants, and 320, mediated through their immunosuppressive anti-inflammatory capability, renders them as promising drugs for arthritis and other chronic inflammatory diseases. As I mentioned at the beginning of my talk, inflammation is very much involved in a lot of medical conditions. So chances are that they should be able to help in many other conditions, and we plan to work on quite a few of these in the next few years, and I hope that many of you will be doing the same. Many of them, that's one of the reasons why THC is not equivalent to marijuana or to hashish, is because it contains large amounts of cannabidiol. And cannabidiol, as you have just seen, has a lot of potency in inflammation. And I believe that there will be some other talks about cannabidiol. So in the few minutes that I have left, I think I have 10 minutes more. Anyway, we'll speak about neuroprotection. We wanted to see what happens to the endocannabinoids during injury. We looked at the endogenous cannabinoids formed by the brain during brain injury. And we saw, as we thought we shall see, that the endocannabinoids, particularly 2-AG, goes up on brain injury. And the red one that you see there, after 4 hours, we get about 10 times more of the endogenous cannabinoid in the brain after brain injury in a mouse. And this could be just a reaction of the brain that has nothing to do with the protection. It just everything is going there in a way that we don't want to, but maybe it's just something that the brain does in order to protect itself, or to try to protect itself from the damage. So we gave a 2-AG, and we saw that 2-AG most definitely reduces the amount of damage. One of the ways to do that is to look at the water, the edema in the brain. Person with brain damage after car accident, for example, the amount of water that goes up is considerably higher. And if it goes beyond the few percent that have increased, normally we have about 78% of water in the brain. If you ask somebody, what are you? He'll say, I'm mostly water. Well, that's true. We are mostly water. But if that amount of water goes up a little bit, from 78% to 82%, it's very, very damaging to the brain. And the neurologist here, Dr. Russo, will certainly tell you, he's a neurologist. He doesn't like his patients to have 82% of water in the brain. Not very good for the brain. Definitely not. And people do all kinds of things in order to prevent that. We found that giving the endogenous cannabinoid will definitely reduce the amount of edema, the amount of water in the brain. It doesn't go down to the normal state, which is 70%, but it goes lower than what we see. So, and we also see the neurological score going down. We gave this compound to these brains to our histologist, and he found that the amount of damage that one can see histologically goes down by between 30 and 50%. You can see the white part of the brain is the one that's damaged in that particular showing, and that's to say that the infarct volume, it goes, it's much lower in the 2AG treated mice than in the control mice. So if we block the antagonist, we block the cannabinoid system, the damage goes up. If we look at the blood-brain barrier, which on brain damage has been either totally destroyed or partially destroyed, we see that the 2AG protects the brain, it protects the blood-brain barrier, which is a barrier that doesn't allow compounds from the blood to go into the brain, because the blood may contain compounds which in very low amounts may damage the brain. Well, during brain damage, this blood-brain barrier is damaged. 2AG, and possibly cannabinoids as well. People have shown that for cannabinoids as well, so have we, and protects the blood-brain barrier. So it works in several ways. Let me say something about something called NF-kappa B. We have a compound in the brain, NF-kappa B, which is there, not active normally. But if there is inflammation, there is brain damage, there is something, this compound, which is together with its inactivator, sits there and waits for its chance. Once there is something that happens, whether it's inflammation, brain damage, or so on, the block of this NF-kappa B is cut out by a variety of ways. Many people working on that very important mechanism. Then this NF-kappa B protein goes into the cells, and these cells start producing inflammatory cytokines and all kinds of compounds of that sort, and therefore the slowing down of the production and free NF-kappa B is extremely important. Now we have just recently shown that 2AG slows down the production of free NF-kappa B. That means that it will slow down the production of inflammation. That has to do with the first part of what I said, but it also has to do with protection of the brain itself, because the two things go together. So we have just sent a paper showing that NF-kappa B production is blocked by 2AG. Chances are that it is also blocked by cannabidiol, which in many respects works in parallel with that. It does not, 2AG is not effective in animals in which the cannabinoid system has been eliminated. There is a biochemical way of doing it. There is a called knockout mice. In case the animals, the system is blocked out, then the cannabinoids have no effect. So it has to go through the cannabinoid receptors, through the cannabinoid compounds. And many other people have looked into that. I see that I have to finish in a few minutes, so we'll not go through the whole literature. This is the summary of this part. I want to say a few more words. The brain injury causes damage through two main channels. One, of which, as I spoke, production of all kinds of compounds. I mentioned cytokines, reactive oxygen intermediates, some glutamate, and so on. These cause neuronal cell death. If we can block that, we have neuroprotection. It also works to action of vasoconstrictors. During brain damage, there is constriction of blood vessels. We can see that the 2AG, possibly also cannabinoids, block that vasoconstriction. And this is something that has not been fully investigated. We have published something on it. A few other people have done so. So chances are that we're looking at two different pathways through which cannabinoids, and we certainly know that about the endogenous cannabinoids, they do that. And a few more